All right, continue on into this second part of this lecture. In the previous portion, we talked about sex linkage, and we looked at an example of an X-linked an X-linked gene. Let's look at a couple of other examples. Table 4.2 of your textbook includes a list of human X-linked traits. Some of those are also observed in some non-human animals. Examples of X-linked conditions observed in humans are red-green colorblindness, hemophilia A and B, and both of those are hemophilia conditions that are associated with clotting factors that are found wherein the genes that encode those clotting factor proteins are found on the X chromosome. There's a disease called Duchenne's muscular dystrophy, which is associated with having a mutant form of the dystrophin protein, and it results in very severe neurological and muscular degeneration. The dog shown there on the right unfortunately has Duchenne's muscular dystrophy, so it does affect canines, and it typically comes on in humans at the age of between 3 and 5 years of age. Other X-linked conditions include X-linked hypophosphatemia, which is also known as vitamin D resistant rickets disease. It's due to a mutation in what's called the PHEX gene. It encodes the PHEX protein, which affects phosphate secretion, and we need to make and metabolize phosphate appropriately in order to have bones that form in a healthy and sturdy way. If you've got X-linked hypophosphatemia, then you have very weak bones and they bow in a condition that is known as rickets. If you are red-green colorblind, then you can't see the number 15 embedded into this red dot circle. Red-green colorblindness is an X-linked recessive condition. It's typically seen in males, okay, because in females, if they carry the mutant allele, it's masked by the dominant wild-type allele, so they don't show the phenotype. So this woman is actually, somewhat rare in that she's showing the condition. She must be homozygous for the mutant allele associated with this condition. She's transmitting one of these X chromosomes to her sons, so that's shown here and here. So their genotype would be this. Okay, they're hemizygous for the X chromosome, and the X chromosome that they have inherited carries this mutant allele, and so they're showing the condition. And then as you look down at their offspring, so if we look over here at individuals III-1, 2, and 3, that male is transmitting only the Y chromosome to his son or sons, and so we can assume that she is either a carrier or she is homozygous for the wild type allele, and she has not transmitted, therefore, a mutant allele to her son. He's unaffected. This is a relatively rare condition, so that's not surprising. We can almost assume that she is X plus in the absence of other information. The daughters similarly are unaffected, even though they are inheriting a copy of the mutant allele from their father. So what we see in these kinds of patterns is we tend to see more impact in affected males, and they inherit the mutant alleles from their mothers, not their fathers. While we're looking at the example of sex linkage and we're thinking about the X chromosome, let's talk about a fascinating topic which is called X inactivation, which means to silence one of the two X chromosomes in XX individuals. In humans, that would mean in women. So this X inactivation is a form of something called gene dosage, or a dosage compensation mechanism. It's a way of making sure that boys and girls, or that females and males, are even or somewhat equivalent in terms of how much X chromosome they have that is functional, so that males and females will have similar levels of the gene products that are encoded by genes on the X chromosome, something called dosage compensation. The process of inactivating the X chromosome is called lyonization, in honor of the woman scientist Mary Lyon, someone who worked in the 1960s. So Mary Lyon and also a scientist called Susumu Ono discovered the process of lyonization. It's associated with a specific transcript, a large RNA molecule that's called the X inactivation specific transcript, or ZIST, or ZIST sometimes. It's a transcript of RNA that accumulates on the X chromosome and effectively shuts off most of the genes on the X chromosome. The X chromosome that has been shut off or lyonized is now can be observed in cells in a very condensed form called heterochromatin, and it's called a Barr body. And the term Barr body is also named after a scientist that studied this process, and his name was Murray Barr. Lyonization and the formation of Barr bodies occurs fairly early in human development, so around day 12 to 16 in human embryos is when this process occurs. So what will happen is here's an embryo, okay, and you will have cells where one or the other X chromosome is randomly inactivated by that large RNA ZIST molecule. So the pink and the blue here are shown, are used by the author to depict whether or not the X chromosome from mom or dad, mom being pink and dad being blue, I assume, although it could of course be the other way around, are inactivated. And so over here on the left, in this cell, it was the paternally inherited X chromosome that is inactivated, and it's just been turned into a little Barr body. And over on the right, it was the maternally inherited X chromosome that was inactivated. Now this is early in embryonic development, right? So when we're just a ball of cells. And so as the embryo develops into a fetus, and cellular replication and differentiation occurs, it means that there's going to be a patterning where there will be pools of cells with one of the X chromosomes active, and other pools of cells where the other chromosome, the other X chromosome, will be inactivated. This results in something observed in females called mosaicism. To be a mosaic means to be patterned. Think about a mosaic or a tile floor. And you can envision it over here, looking at this cartoon of female women. Okay, so what will happen is if you look across the body, there will be patches or areas of the body where you have cells that contain the paternally inherited X chromosome in an active form, and other patches where it's the maternal X chromosome, the maternally inherited X chromosome, that's in an active form. And so if the genes on the X chromosome of that organism were found, if you had two different alleles, if they were heterozygous, you're going to see this patchwork effect. Over here on the left, we have a cartoon that's explaining how this mosaicism due to X inactivation results in calico cats. So calico cats are almost always female, and they are the result of mosaicism. A calico cat, which is female, is going to be XX, and she's going to have inherited X chromosomes from one parent that carry the dominant B allele for a gene that controls color formation, and the other X chromosome will have the recessive B allele. The dominant is associated with orange, and the a little b recessive is associated with black. And so in some of her cells, the X chromosome that carries big B will be inactivated. That's a Barr body shown there. And in others, it'll be the X chromosome that carries the little b. As those cells replicate and form, pools of cells that are distributed over the surface of this cat, you'll have patches of tissue that produce black and patches that produce orange. And that black-orange patterning is referred to as calico in cats. So when you look at a calico cat, you're looking at the result of mosaicism due to X chromosome inactivation, and you're looking at that inaction. These pathways depict what happens in males and females where you have only one of the B alleles because either the cat's male or heterozygous for that particular gene. The sex of an organism can significantly affect its phenotype in a variety of ways. When the impact of a particular genotype is reduced in one sex versus the other through differences in gene expression or anatomical and physiological differences, that's referred to as having a trait that is either sex-limited or sex-influenced. And examples would be lactation and also male pattern baldness. So lactation, or the ability to produce milk, is due to genes that are found in both male and female mammals. However, lactation typically only occurs in female organisms. So there you have a sex-limited trait. Male pattern baldness is also something that is typically only seen in males, regardless of genotype. So the sex of an organism then, in the case of male pattern baldness, limits or influences the baldness trait. And this particular table depicts genotypes and then corresponding phenotypes and how they play out differently in male versus female organisms. And this is humans that this is referring to. Right, there is a wonderful online tutorial found at the website included in this slide. I'd like you to go and view and take notes on that tutorial. It describes some genetic qualities referred to as penetrance and expressivity. There is an associated interactive quiz that is graded online. You just have to click around and you'll see if you've got the correct answers or not. Please take the quiz as well as viewing the tutorial, and know that a question from the quiz will be on our next in-class quiz. The phenotype of an organism is not always immediately known just because you know its genotype, and we've already looked at a few examples of that. Let's think about a specific example where environment can modulate phenotype or modulate the impact of a genotype. The disease called phenylketonuria, or PKU, was discovered in the 1930s by a Norwegian physician. His name was Dr. Asbjørn Fulling. And it's something that scientists of the day referred to as an inborn error of metabolism. We'll talk more about that going forward. And here we see again tyrosine metabolism, something that we've got a lot of examples so far in genetics class looking at the amino acid tyrosine. So you should know from that just how essential tyrosine is and how important it is that it's metabolized properly in the body. So PKU is a disease that is autosomal recessive. It's something we test babies for at birth. So it's one of the very first blood tests that is done on an infant, brand new infants. They do a heel prick, they collect blood, and they test them for PKU and a variety of other things. It's really dangerous because if you don't treat it, the infant will develop into a child and an adult that has very severe mental retardation, very delayed overall development, and tend to have very short and unhealthy lifespan. And the good news is that it can be managed very effectively by diet. So PKU is due to having a deficient tyrosine, a deficient phenylalanine hydroxylase. Okay, so look at this step in the pathway. Okay, phenylalanine is an essential amino acid, and it is in most of us metabolized to tyrosine. Okay, and then the tyrosine is metabolized to dopamine or other metabolites or products, for example, eumelanin. So remember that biochemical pathways exist in our cells in basically a web that is very interconnected. So this is just one possible fate for phenylalanine and tyrosine. Dopamine, as you should know, is very important in normal neurological function. So in order for our nervous systems to function properly, we need appropriate levels of dopamine. It's a neurotransmitter. We get phenylalanine, which is an amino acid, from our diet. Okay? If you have PKU, then you cannot properly form phenylalanine hydroxylase and metabolize your phenylalanine to tyrosine. And what will happen is the phenylalanine will accumulate, and then because it's accumulated, it'll be metabolized in different pathways that lead to a variety of problems in the organism. One of the problems, though, is that you'll see reduced levels of dopamine, and that's the disease called phenylketonuria. The fact that you can manage this by diet, and the way to do that is low proteins, like have a diet that has no phenylalanine, okay? No phenylalanine essentially whatsoever in it. If you don't consume phenylalanine, then you don't experience PKU as a disease, and you can develop somewhat normally. So you can manage this disease by managing the environment of the organism, and the environment of the organism in this case is dietary management. You're managing the biochemical environment. Please go on next to the last and final part of this lecture.